Hello, I am Saurabh Mukherjee and I am from Kalyani University, Kalyani, Noida. So in this video, I am discuss about the protein and ligand interaction and protein interaction, which was also called the molecular docking. So basically, the molecular docking is a computational approach which is used to the predict to the orientation of a ligand. So it is also a powerful tool for computer-aided drug design and it is a part of the structural biology. So the molecular docking is composed of or molecular interaction occurred between the protein and ligand, and the second one is protein. So this molecular docking approach is occurred between the protein and ligand and protein to protein. So the interaction occurred between the protein and ligand and protein interaction. So it is this interaction used to predict the preferred binding mode of a ligand to a protein or protein to a protein. So in the next slide, uh, I'll discuss about the molecular docking process between the ligand and the protein. So this is a slide about the general overview. So first of all, for molecular docking, we required a protein which we get from the PDB website, that is protein data bank, and the ligand or the drug which I found from the PubChem or drug bank or many other website from where the structure we found in the PDB format or SDF format. So if we found the structure, suppose we found the structure, the ligand structure in SDF format from the PubChem. So this structure is further transferred to the or further transformed to the PDB format via many other tools. That is one is most common tool used to change the format between the molecule, that is open bubble, a common or most useful tool. So after that, after we getting the structure uh, of ligand and protein, the after the getting the structure of ligand and protein, we need to require this protein and ligand to prepare. So prepare in preparing in Autodoc application or Autodoc network. Next, define the binding site for docking stimulation. So binding site, for define the binding site, a grid box is required. In case of blind docking, we need to cover the whole protein, but in case of site specific docking, we need to require the cover the specific site. So next, after grid box preparing in Autodoc, the docking stimulation run or perform simulation run or perform in Autodoc or Autodoc Vina. So most updated tool is Vina. Autodoc Vina. Autodoc Vina. It is a most user-friendly tool and used some command to perform the molecular docking, and at last we found the result, and between the ligand and protein. Further, the results are analyzed in PyMol or in Discovery Studio or many other tool or software is used for analyze the result and also the structure prediction among the various process which were found after docking. So this is the general overview of protein and ligand interaction. And next, so this is all about the ligand protein interaction, that is the molecular docking in ligand and protein. And protein protein interaction I will discuss in later, and we are found the protein protein interaction video or lecture in the next video. So thank you, best of luck.